All right, I'm Summer. This is
Annie.
and
Theo.
all right, we are Table D from Sideburns, and this is our bacteria research project. Our question was, how will fruit juice affect bacteria growth over a 2-day period of time in an incubator? Our hypothesis was that we thought that the fruit juice would have much less bacteria growth than our negative variable water. We thought this would happen because of the fruits all have a low pH level. So we didn't think that bacteria would grow very well in it. All right, here's our expected graph. So it shows that the lower the pH level, the less bacteria growth or percentage of plate that the plate will be covered. And 2.8 is pomegranate, 3 is blueberry, and 3.5 is orange, and then 7 is water. Here's our research paper.
Okay, so we all researched the different fruit juices. Summer did pomegranate, I did orange, and Theo did blueberry. And then we combined them in this research paper, but these are the most important facts that we found. So Summer found that pomegranate juice stops bacteria from multiplying as well as killing it. Theo found that blueberry juice kills many harmful bacteria, and I found out that pH level is a big variable in whether or not bacteria will be able to grow in the fruit juice.
Okay.
This is our bibliography.
Yeah, and this is the materials.
For materials, we need the Q-tips to swab the plates and get the samples of bacteria and juices. We need the Petri dishes to have a place for the bacteria to grow. Pomegranate, blueberry juice, and orange juice for the samples, and parafilm to seal the plates and make sure no bacteria gets in or out.
Okay, so the way we scored, took the amount of bacteria, was after the plates were in the incubator for 2 days, we took the plates out and we looked at each half of the plate and we decided what percentage of the plate about was covered with bacteria and the procedure.
First we need to get the materials, then we need to get the bacteria from my mouth, get the samples from the juices, streak the dish like below, down here, and repeat the steps with all the juices, and then do 3 trials.
Okay, here is our data table.
So this is where we put the percentage of the bacteria. You can see that the orange pH level, the pomegranate, and the blueberry, as well as the water. But you might be able to see it better in the graph, which is below, and someone will explain it.
Okay, so on the y-axis we have the percent of the plate covered, and then on the x-axis we have the pH level of the different fruits. 2.8 is pomegranate, and then 3 is blueberry, 3.5 is orange, and 7 is water. So our data turned out much different than we had expected. So each line represents a trial or the average, and for pomegranate, everything was 20% of the plate covered or below, which is about what we expected. But then we expected blueberry to be around 40. Orange to be around 60, and then water to be around 70, and that didn't happen at all because the other juices, blueberry and orange, were much higher, and water was lower or as low or lower than pomegranate, so it was much different than we expected. Here are the pictures.
As you can see, these are the pictures for trial 1, trial 2, and trial 3. We put the pomegranate and water on the same petri dish and blueberry and orange on the same petri dish, although on opposite sides. And as you can see, blueberry and orange had a lot more bacteria than pomegranate and water.
Okay. So before our data turned out much different than we expected. We think that this might have been because of the fact that the blueberry and orange juice had not a lot, but a small amount of pulp that was probably transferred onto the dish, and that may have affected how much growth that, and so that was one of the things that might have messed up our data a little bit. So if we were to change it, we might change that next time. But yeah, that's